DNA technology. So I'll be talking about gel electrophoresis. It is a technique for separating molecules based sa ilang size, and it's particularly used with DNA. So this is used to visualize genomic DNA, RNA, PCR products, plasmid, restriction, enzyme digest products. So the gel used in electrophoresis is usually made of a material called agarose. So this substance is extracted niya siya from seaweed. So the materials used: casting tray, a gel, a comb, electrical supply, DNA sample. So how does it work? First, pour agarose gel into casting tray with comb. This is the comb, and allow it to solidify. So you will put it here as a negative terminal to form a well-like structure like this one, pahuman. But since it is colorless, we will put a colored loading dye here later. And then, but before that, we will fill the apparatus with a buffer. This is used to provide better conductivity of electricity. Next, then the DNA mixture will move through the gel. So this is the dye. Colored na siya. So makita na dye na siya. So the DNA. So the results. Dayan. DNA band. So when DNA is observed under ultraviolet light, bright colored that are orange bands are clearly seen. So these are nothing but the bands of the DNA. The separation has been successful based on the size of the DNA. So the larger molecule are the ones na makitaan here, which means kani ang naglihok o dugay. On the other hand, kaning mga gagmay na molecules ang nag-move fast. DNA ladder. So this is how we would know the size of any fragment, the DNA fragment. So the DNA bands are obtained and compared here with the standard chart known as a DNA ladder. So kung mabala na to ang length sa mga fragments, ma-identify na nato easily ang desired DNA. Elution. Now, kanina DNA fragment can be used manually, cut out from the agarose gel kanina, and then extracted. This process then is called elution. So kaniyang it's extraction of the DNA. So it is done in such a way that we can be used to further downstream processing, as it is said here. Visualizing DNA, ethidium bromide binds to DNA. Fluorescence under UV light makes bands visible. So the uses of electrophoresis is to separate DNA fragments for DNA, fingerprinting to investigate crime scenes, to analyze results of polymerase chain reaction, to analyze genes associated with a particular illness, and many more. Pero kaning electrophoresis is often used na siya para mag-determine og related na sa lahi-lahi na mga species. So makatabang siya sa mga scientists para or other people na para ma-identify nila ang mga organisms. So part po din siya sa DNA printing. So like kaganina dere, one of the examples, if you have a DNA sample from a crime scene, you can go through the steps of gel electrophoresis para ma-compare nimo ni sa DNA sa suspect para makita nimo kung gamatch good ni siya dito sa inyong ginapangita. So that is all. Thank you.